Inside almost every cell of every plant on Earth, chloroplasts are working hard. A chloroplast is a plastid, a tiny protein-filled unit within the cell, responsible for the vital process of photosynthesis that provides us with food to eat and air to breathe. But that's not all a chloroplast does. In fact, these minuscule machine-like plastids perform all sorts of functions, aided by their remarkable plasticity or their ability to change, and they are critical to a plant's ability to respond to environmental factors like light or the threat of disease. They do this by importing different proteins from elsewhere in the cell to equip them for new conditions and new functions. Research scientists at the University of Oxford wanted to better understand how plastids in plants regulate levels of different proteins to ensure the efficient functioning of the plastids themselves, the cell, and the plant as a whole. They discovered that as well as importing the proteins they need, plastids can also remove the proteins they don't need to refine their functions and improve their efficiency. They called this removal system CHLORAD, which stands for chloroplast-associated protein degradation. At every stage of a plant's life from the moment of germination, CHLORAD monitors and regulates proteins to optimize the plant's success. The researchers believe it will be possible to control and manipulate the CHLORAD system, allowing them to fine-tune plastid proteins and boost crop plants' abilities to produce nutritious food. They could create wheat that stays green for longer to produce more grain, improve the nutritional value of a potato crop, decrease the time it takes for a tomato to ripen, or keep it fresh for longer. And that's good news for farmers, and good news for a hungry and growing world population. For more fascinating science, visit our website, subscribe to our channel, and like us on social media.